Hello students, welcome to EPG Pathshala. I am Devapriya Chaudhary from Jawaharlal Nehru University, and today we are going to continue our discussion on pattern recognition methods and sequence analysis, which is of course under the paper Computational Biology. So, as we had discussed in the previous module, several methods of pattern recognition. We went through sequence motifs, we went through position-specific weight matrices, then we moved on to psi-blast, and now I'm going to discuss a particular method, a class of methods called the Markov models, and their use in sequence analysis and sequence searching. To understand these Markov models, let's start with a simpler representation called a Markov chain. Let us imagine you are tossing some coins. Now at every toss, you can get head or tail. Let's say with some probability you get head and some probability you get tail, and then you toss again. Now each toss, let me call it a state. Now in that state can have two values. In our case, our example, head or tail with some probabilities. So these probabilities, the probability of getting head or probability of getting tail, will be the emission probability of that state. And then from one toss to the next toss, you decide whether you're going to toss or not, also with some probability. So we will say that one state is related to the next state by a transition probability. So in any sequence of events, if I divide the set of events into discrete states, and each state can have different outcomes with a set of probabilities, the probabilities of these outcomes of each state will be the emission probability, and the probability that you will move from one state to the other state is the transition probability. Now, it may be a simple sequence. In that case, it's called a Markov chain. Or it can be that from one state, you can go to state this one or a state this one. In that case, you have separate transition probabilities related, and you do not have the chain like this pattern anymore. In such cases, we'll call it a Markov model. Again, let me reiterate, the Markov model is characterized by a set of states which are related by transition probabilities, and each state has a set of outcomes, and the probability of each outcome are called the emission probabilities. So I have a set of states and I have a set of two kinds of probabilities, the transitions and the emission. Together I get my Markov model. Now the key property of a simple Markov chain of order n, that is I have n states in that particular chain, is that the probability of each symbol xi depends upon the preceding symbols, n symbols which is there. Say for example, if I can define that whether I would get head in the fourth toss or not depends upon whatever I have gotten in the third toss, in the second toss, in the first toss, etc., then such a kind of sequence of events can be modeled by a Markov chain. So now let's look at it a little bit more mathematically. Let's consider the probability that a particular sequence is obtained. So we'll call it P of x, and which of course depends upon P of x at L minus 1 until the first one. That is, the probability that I am in a particular state emitting something would depend upon the probability that I have visited all the previous states thus far in a certain order. So this I can write as a conditional probability, that this probability Px now depends that at the Lth state I've got x, given at L minus 1, L minus 2, L minus 3 until the first state I've gotten some symbol, right? And times the product that at L minus 1, whatever probability I've gotten, given that whatever I've got at L minus 2, L minus 3 and so on and so forth. Now, writing this complex equation, let me remind you now of something called the Bayes formula, which is given as the probability of joint probability distribution of two events, Pxy, is given by the conditional probability of P probability of x given y times the probability of y itself. Applying this, the Bayes formula, to the previous equation, right, I can now go further. Let me make one more simplification. This long thing, probability of xi given probability of xi minus 1, let me write it in a shorthand notation as axi minus 1 times axi. So given that, the previous equation now reduces to a simple product of probabilities, which is given as Px is now Px at position state L is the product of whatever this probability is, A, x, I minus 1, A, x, I, for all I is equal to 2 to I is equal to L, all the states. So it's the product of all of these different probabilities. Now these conditional probabilities, Pxi given Pxi minus 1, can be estimated from the observed counts, which I measure from by analysis of different Markov-like phenomena. For example, if I'm looking at a nucleic acid sequence, for example, I can always estimate the Pxi given Pxi minus 1 from the observed counts of the, say, dinucleotide frequencies. So I can extend the same formalism to higher order Markov chains. So if I want to make a third order Markov chain, so I have to count every trinucleotide frequency and so forth. So in this case, additional conditional probabilities needs to be estimated. And so with this, I can create for at least a DNA sequence or even a protein sequence, I can create a Markov model of any order for that matter. So here we call it the n-1 order Markov models. Now what use are these? The use of Markov models or Markov chains can be used to discriminate between two kinds of situation. Now let us say I have one sort of situation, I define it with a Markov chain. I have another sort of situation, I define it also with another Markov chain. Now my event can take any of the two paths. Now what is the probability that it will take one path more than the other? If I can estimate this for this individual Markov chains, I can define a log odds ratio, which we have seen before, simply using the equation given out here. S of x is log of Px given Markov model 1 divided by Px given Markov model 2, and which in terms of the shorthand notation I have said becomes a summation of the beta xi minus xj, where the beta xi minus xj itself is nothing but the probability of xi given xi minus 1 and so on and so forth. So this sort of log odds ratio can be fairly easily defined, which then tells me the probability that I either in this particular state, this particular sequence of states, or in that particular sequence of states. So where do we use this kind of concepts? One of the places where this is used is locating a gene. Now why do I have to locate a gene? Nowadays it's common practice to sequence the genome of an entire organism. Now if we take the human genome, which is a huge sequence, right? Segments of it are actually protein coding genes. Much of it are so-called junk DNA or DNA sequences with no obvious function. Now, if given such a large genomic sequence, I need a method which is most likely going to identify to me those functional regions which either code for protein or code for some regulatory element, some RNA or whatever, right? And these are the likely genes. And how do I do it? Okay, now one way to do that is that if this particular gene in this organism of interest matches the sequence of some particular gene which I know exists in another organism, I could use a homology based method like, say, for example, BLAST or SciBLAST. But what happens if there is no such homology? I get an organism which is so strange, so rare, that its genes do not match with anything. What do I do? So one of the things that I can still do is analyze with the help of Markov model. Let us see one of the uses of fixed length Markov models. There's a program called GeneMark which finds genes from an unknown sequence using a fixed length Markov model. Now why does one do that? Now many times when you have sequenced a genome, you would like to see if there are any genes which are present there which you know about. Now of course one way to do it would be do a BLAST search and see if it is similar to some other gene. But that may not always be possible, because there might be some gene which is so different from others that there isn't any homologous sequence already existing in a database. In such cases, programs like GeneMark can be used. It uses a fifth order Markov model. That means what it does is that it has in a training set, it has created the frequencies of all the 4096 pentanucleotides. So given a gene sequence, it looks at all the possible pentanucleotides which are present in your unknown sequence and tries to see if their occurrence is predicted by the Markov model which has been generated using this 4096 pentanucleotide transition frequencies. Now, while this might work, there are certain problems out there. Now think about it. There are only 4 single bases. There are 16 dinucleotides. There are 64 trinucleotides, 2056, 256 tetranucleotides, and 4096 pentanucleotides. Now, what would be the length of the DNA sequence database which is required to give me an adequate representation of these bases or dinucleotides or trinucleotides? Obviously, since there are 4 bases, if I have a database of, let's say, 100 nucleotides or so, I will get a fairly good idea about the relative frequencies of these 4 bases, at least up to 1% accuracy. On the other hand, if I look at dinucleotides, there are 16 of them. So, in a random sequence, if all of them are equally represented, just to get one dinucleotide, I must have at least a 16 mer of DNA. Now, when I have so large 4096, it may be that my database is not of sufficient length, which gives me good enough counts of each of the 4096 pentanucleotides to be able to accurately construct the transition probabilities. So, which means small training sets, they cause problems because the k nucleotide frequencies or counts are not sufficient enough to give you a reliable estimation of the transition probability, that's their frequency. So what does one do in such case? So higher order Markov models, of course, can perform better than lower order. That is, any model which works at, say, 5th order frequencies generally performs better than 4th order or 3rd order or 2nd order. However, if your training set from which you are training this Markov model, you are obtaining the frequencies, the probabilities, if that's not large enough, then you cannot reliably create a high order Markov model. So what do you do in such cases? So wherever possible, use as high as you can go. Wherever not possible, you come down lower in the order. So for example, if for 4096 pentanucleotides, my database is as a reasonable representation of, let's say, 4000 of them, so then I use it. But for the remaining 96, my database is not good enough, it's not big enough. So what I do, I move down to a 4th order Markov model. Now there are only 256 possibilities in 4th order. So a database which works well for a majority of pentanucleotides definitely is likely to work better for tetranucleotides and so on and so forth. So I come down one step below. Such kind of Markov models which are not fixed in their order, they go up to a certain level whenever possible, and then they come down a little bit whenever that thing does not happen. So this kind of Markov models are known as interpolated Markov models. And there are a lot of gene finding programs based on interpolated Markov models. So in these ones, we use higher order statistics wherever there is sufficient data, but fall back to lower order statistics. So here is an example of an interpolated Markov model of, say, kth order. So in this case, my frequencies are lambda k wherever it's available. When not available, I use by some certain weight, I use a lower order. So I am a k minus 1, right? So in this way, the lambda k is a weighting function. So if possible, if I have enough statistics, I use the weighted function for the highest order possible. If I do not have, then I use a 1 order lower, right, for wherever it's not possible, and I can go on recursively in this one. So that way I can go on as many orders below that I have to go. But the lambda k sx minus 1 is a numeric weight, and this weight also needs to be trained. So the probability that a sequence is generated by a model is now given ps, the sequence given generated by the model M is given as the scores obtained from this interpolated Markov model. So, and it is a summation of the scores for every possible k length window. So therefore, the kth order Markov model is just a linear combination of its lower order Markov models. So one example of an interpolated Markov model based program is called Glimmer, and it's used for gene finding. So when applied to several bacteria, say Haemophilus influenzae, it finds 1738 genes which are known to be there. 1720 of them are found out by Glimmer, so gives you a 99% accuracy. In most cases, as you can see, that the accuracy is between 97 to 99%, which is, I should say, a pretty good approach. So interpolated Markov models work slightly better than fixed order Markov models. Now, but even these have a little problem, because here we are on the base of an assumption that I am looking for a gene which has a certain pentanucleotide or tetranucleotide frequency. Based on that, I've created my Markov model. But sometimes, even in your training set, you don't know where the gene is there or where it's not there. Then how do you even construct such Markov models? An example comes here. Say, for example, in the human genome, there's this dinucleotide CG, C phosphate, G cytosine, phosphate, guanosine, or we'll call it CpG. Is usually, whenever it occurs, the cytosine nucleotide is typically modified by methylation, so we get 5-methyl cytosine. Now, 5-methyl cytosine has a strong chance of getting mutated to thymine. Hence, you find the CpG sequence is rather rare, because it would have gotten methylated and then it would have mutated itself into thymidine. As we have seen, the interpolated Markov models are fairly good for certain things, like for example gene finding. But there are certain complicated situations. Let us imagine a situation where even in the training set I cannot really identify where the gene exists. How do I then train it, an interpolated Markov model, to find such kind of genes? To give you an actual biological example, I'll take the case of the so-called CpG islands. Now what are these? Now in the human genome, whenever this dinucleotide CpG, cytosine followed by guanine, occurs, the C nucleotide is typically modified by methylation at the 5 position to 5-methyl cytosine. Now 5-methyl cytosines, they have a strong chance of getting mutated to thymine. Hence, it is relatively rare to find the CpG dinucleotides occurring inside the genome, right? So generally you don't find them, because wherever they occur, chances are that the cytosine has gotten methylated and has mutated itself to thymine. For certain good enough biological reasons, which we know now, this methylation of cytosine is suppressed near certain important regions of the genome, namely in around promoters or transcription start sites. As a consequence, in such kind of regions which are promoters and so on and so forth, the frequency of CpG dinucleotides is rather higher, and these are called CpG islands. And typically CpG islands are only about few hundred to a few thousand bases long. Now it is not to say that within the island you only have CpG, you have also other nucleotides. And it is not to say that outside of an island you don't have CpG, you have. But it just so happens that within the island, the frequency of the CpG dinucleotides are relatively larger. As a consequence, CpG dinucleotides are more common in this island regions. Now this is not to say that within a CpG island, you will not find other nucleotides. Yes, you will do. And it is also not to say that those parts which are not CpG islands, you will not find CpG. Yes, you will. It just means that there are certain regions in the genome where the frequency of occurrence of CpG is a little bit more than certain other regions. And typically these islands are about few hundred to few thousand bases long. Now because most of the time the CpG islands occur in regions which are important, promoters or transcription start sites, it becomes very important to locate these islands accurately. So we have the problem of identifying the CpG islands in a long unannotated sequence. One way to do this would be to set up two different Markov models, one for CpG islands and one for non-CpG islands. The same way we do that for gene finding. We set up one Markov model interpolated Markov model for genic regions, which we know, and we try to create another Markov model for non-genic regions. And then given an unknown sequence, we run it through both, we calculate a log odds ratio and see if it is above a certain threshold. Yes, we can do that here as well. However, there is a little bit of a problem. So again, let me reiterate. So given a text sequence, any test sequence, we run it through two separate Markov models, one for CPG islands, one for non-CPG island, take the log odds score and get this log odds score S. And one can then use this discrimination score in a sliding window of say 100 nucleotides and then plot it, and whenever this discrimination score is higher than certain threshold, we claim that this is a CPG island. But this approach has a little problem at the boundaries between CPG islands and the non-CPG island is not clearly identified, because if your window length is rather big, and as you are sliding your window, as you move from the boundary of CPG to non-CPG, your score is slowly, in a smooth fashion, it is coming down. So this does not allow you to identify the boundaries. And most of the time, that is very important. So the length of the window reduces the sharpness of the signal. So what do? One approach is now to build a single Markov model for the entire sequence, which contains both this Markov chain, one for CPG, one for non-CPG. So given two Markov models, Markov chains rather, one for CPG islands and one for non-CPG islands, I can define this discrimination score, which is a log odds score. So I find out the probability that a particular sequence belongs to a CPG island or it belongs to a non-CPG island, take the ratio, take the log, and I get this score. Now I can calculate this score in a window, and then I can slide this window looking for the value of the score. In a CPG island, it should be rather high. In a non-CPG island, it should be rather low. And as I pass from a CPG island one to the other, it should come down, as it should remain up and then it should come down. Unfortunately, because this window length is rather large, what would happen? At the boundaries, it is not going to come down very sharply. It's going to come down rather slowly, which would mean that I have a difficulty in identifying the boundary where one CPG island ends and the non-CPG island region starts. Now what can I do for this? One approach would be to build a single Markov model, not two separate Markov chains, Markov models for CPG and non-CPG, just a single Markov model for the entire sequence, which incorporates information both from the CPG islands as well as from the non-CPG islands. However, using this sort of a combined approach, there is one complication, that earlier I had this one symbol, one state. That is, either A, T, C or G, which are the symbols, they either belong to CPG or they belong to non-CPG. There was no way they could have mixed up. But now that I am having both the chains incorporated in one, I have a situation. I might have an A, C, G or T, which may be part of a CPG island, or the same things may be part of a non-CPG island. So one way to avoid this problem, that I define two kinds of bases. I define A plus, C plus, T plus and G plus, which are the same thing, adenine, cytosine, thymine and guanine, when they come from a CPG island. Or I also define A minus, C minus, T minus and G minus for the same four bases, but this time coming from a non-CPG island. So this is the overall shape of my Markov model. At the bottom I have the non-CPG islands, A minus, C minus, G minus, T minus. At the top I have A plus, C plus, G plus and T plus. I have transition probabilities for every state going to every other state. Now, given a particular base, X, it is no longer possible in this kind of model to just identify whether it is coming from a CPG island or it is coming from a non-CPG island. So in this kind of situations, the underlying states, which is emitting the sequence of symbols, is actually invisible or hidden from the observer. And hence, this sort of Markov model, we will call them hidden Markov models or HMMs. Now, whenever we are dealing with HMMs, we can ask certain questions. Now, what are these questions? So one of them is like this, that take an observed sequence O, which is made up of a sequence of symbols O1, O2, O3 till ON. and I have an HMM lambda. Now it has its own parameters. The emission probability is A, transition probability is B, there is a starting probability pi, and so on and so forth. So I have an O, which is the observed sequence made up of symbols O1, O2, O3 till ON. I have an HMM, I define it lambda, which has its parameters A, B, and pi. So the problem here is, how can I efficiently compute the probability of observing the sequence O given the hidden Markov model lambda? So given a sequence and a hidden Markov model lambda, I simply ask the question, what is the probability that this sequence could have been emitted by this hidden Markov model? Now this kind of a question or a problem, we will call it the evaluation problem. The second question which you can ask is that, again, let us take an observed sequence O made up of symbols O1, O2, O3 till ON, and again I have a Markov model lambda with parameters A, B, and pi. Now I can ask this question now, that can I determine a state sequence that is a sub part of my hidden Markov model, right? Say, which I'll define as Q with the state sequence Q1, Q2, Q3, which that sub part best explains the occurrence of the observed sequence. So I might have a very complicated hidden Markov model, but I'm asking, is there a subset of that hidden Markov model? Is there a Markov chain within it which is best capable to explain with the largest probability PO lambda given my observed sequence O, right? This is called the analysis problem. And then finally, the first two problems will even occur only after I have had created a Markov model, right? So the last question I'm now bringing, it should have been perhaps the first question, is how do I train a Markov model? That if I have a lot of sequences and I want to create a Markov model lambda, how do I do it? And that of course is the training problem. So in the next few slides, I will try and tackle each of these questions in order. So let's try to solve the evaluation problem. Now let me define the probability of having the observed sequence given by a hidden Markov model lambda, which has the following state sequence I, again comprising of I1, I2, and IT. Now this probability PO given lambda is given by the summation of the probabilities of P given O given I and lambda times the probability of I and lambda itself. We further defined an initial state, let me call it pi I, and for which the probability is pi. Okay, so a starting state which is not part of my sequence. And I defined a transition probabilities Aij, which is given the probability that I will move, given state I, the tth state is I move from t plus 1 state will become j. So, and again I define a parameter b, which says is that what is the probability of observing the symbol vk if we are in a state j, that is the emission probability. So again I have a starting probability pi, then I have a transition probability aij, and I have an emission probability b or bj. Now given all that, now it turns out that the probability I'm looking for is given by that long equation, summation of pi times b times O1 aij to t blah 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 blah. Now this is a fantastic equation, easy to derive. It turns out that this is extremely difficult to calculate. So if you have n states, n possible symbols in t different states, then to evaluate that equation you will need n to the power t calculations. Now that is really too huge. So in practice this direct solution method is not used. What we have instead are two algorithms, basically the same algorithm, how you look upon it. One is the so-called forward algorithm and the other one is dual, the backward algorithm, which simplifies this equation and gets you that probability in a lesser number of steps, which make it more easier to calculate. So in general, whenever you hear the evaluation problem, what is the probability that this sequence belongs to this Markov model? The answer would be forward algorithm or its counterpart, the backward algorithm. You can use any one. So that algorithm is what we are going to use to obtain that probability. Now if you look at the analysis problem, Here my question is slightly different, that I have a Markov model which has a certain state sequence within it, that I is equal to I1, I2, I3, and so on and so forth. Okay. Now, if I have an observed sequence, O is equal to O1, O2, O3, and so forth, so and so forth, the question is, how well does the state sequence I predict the observed sequence O? So, which means that I am asking the question, what is the joint probability of having the observed sequence O and the state sequence I given my Markov model? Again, the most popular way of doing this is called the Viterbi algorithm. I will not describe it much, the mathematics behind it, excepting to say that it is a kind of dynamic programming algorithm which you have seen when we are doing Needleman-Wunsch and Smith-Waterman algorithms during the alignment section. So it's something somewhat similar to that. We have this Viterbi algorithm which finds out the joint probability of my observed sequence and some state sequence I within my Markov model. And what I'm interested is in finding the state sequence I. And then of course the last and most important thing is that how do I construct my Markov model? So this is the problem which trains the HMM. You give it a lot of data as part of a training set and then you create the HMM from nothing. Now the way to do it, we have an algorithm called the Baum-Welch algorithm, or sometimes also known as the expectation maximization algorithm, which is an iterative algorithm which starts one step at a time and repeats that step many times until convergence, and which is then used to create a trained HMM. So again, summarizing the situation, what we have, that we have these Markov models which essentially gives you a probabilistic view of a certain event, which may be occurrence of a sequence or maybe so many other things. Now, given such a Markov model, we can ask a few questions, that our sequence or our event, what is the probability that it can be predicted by a particular Markov model, so-called evaluation problem? Answer to that is the forward algorithm. The second question we can ask, that given this particular sequence fits this particular Markov model, is there within that Markov model a certain state sequence I which best predicts my observed sequence? So basically, I'm breaking apart my Markov model and finding out something from inside it. So we will call it the analysis problem. Answer to that, the solution to that is the Viterbi algorithm. And finally, the last question is that, okay, how do I get my Markov model? So how do I train it? Given certain training data, the method to train that is the Baum-Welch or the expectation maximization algorithm, and that can be used to train a Markov model. So these are the three problems and there are three solutions. So while the hidden Markov models, which I've been describing thus far, have come a long way, they are really very powerful methods. Now, for biological sequence analysis, a little bit simplification can always be obtained, something which we call the profile HMMs. Now again, we know that biological sequences often come in families, and the purpose of many a sequence analysis tool is to uncover the relationship between a particular sequence and its own family, or to discriminate between different families. That's what we have been talking about with our multiple alignments. We have been talking about the same thing with our profiles or PSWMs or whatnot. So if one already has a set of sequences belonging to one particular family, and one can do a database search with this family, we can uncover more members of that sequence family. And to do this, again, a kind of Markov models called the profile HMMs can come in quite handy. So again, we have seen this before, that a multiple alignment captures many characteristics of a sequence family, and if you can take the entire Markov multiple alignment and use it as a database search, it would be a pretty useful thing to unearth more members of that family. Now just if we look back a little, that we see that our sequence motif, essentially what it does, it condenses a Markov model, multiple alignment into one motif. If we look at our weight matrices, what it does, essentially takes a multiple alignment, calculates the frequencies, and tries to represent it. Now, in case of our motif, we had the problem that just amino acids are present there or not there. That's all we have. we do not have their frequencies. In case of the weight matrices, we do have the frequencies, so in that sense it's better than the motifs, but it has a problem. We cannot handle gaps. Now we bring in the HMMs, which would be just like PSWM, but at the same time now we will try and model gaps. So again, these three things, the motif, the PSWM and the HMM, all of them take as their input a multiple sequence alignment, but they treat it at different level of detail. At the lowest, I have a sequence motif, very little detail. Somewhat more detail is a PSWM, okay, HMM with a lot more detail. And how is it that it's handling both the amino acid frequencies within the alignment as also the gaps, etc. This is what I'll see next. So let's start with a gapless region of a multiple alignment and let us try to make it a probabilistic interpretation. So again, I can define the probabilities EI of A for observing amino acid A at a position I in the alignment. So the probability that a new sequence X which conforms to this model, that is Px given n, will be the product of all of these EIs for all of these positions that I have gotten. So that's the equation over there. Now the ratio of this probability that X belongs to a particular family as opposed to X belong to a random model, again I can use a log out score and I can define S is equal to log of EI over Q, and instead of the product, since it's in the log scale, now it will become a summation. So I have this particular formula. Now these values, the log of EIA over QI, is nothing but the PSWM, right? So I can call it nothing but a score matrix SA of B, where the second index is the position rather than the amino acid. So these things we'll call it the position specific scoring matrix. Now a PSSM can be used to search for a longer sequence X in a database. Again, the function of this is exactly as a weight matrices operate. Everything is there because it is still not handling gap. So if you can now upgrade the PSSM a little bit to handle gaps, then I go one better. So one way of doing this, of course, is using it in Markov models. How do I do it? So I can view my PSSM as a very simple HMM, which is just consisting of a sequence of states. So I have a begin state, and then I have, for each position in my PSSM, I have a particular state, A, B, M1, M2, M3, Mj, and going and ending in a particular end state. The emission probability of an amino acid at each of these states is simply the frequency of amino acids at that particular position of the PSSM, right? That is fine. So how do I now, to handle gaps, indels, I can think of them as handling insertions and deletions. I have to add additional states. So we model insertions and deletions as new states. Okay, for insertions, I will, between a state I and a state j, I will now create a state k in between them with transition probability going from I to k as well as I to j and k to j. Again, from the state mj to mj plus 1, I have added an insertion state. Now from mj you can go either way. So in some of the sequences from mj you will go straight away to mj plus 1, but in some of them from mj you will go to ij and then move from ij to mj plus 1. So where you are going via ij, you are having an insertion. Where you are going direct, you do not have it. So deletions are handled in this similar way by, now we will have some silent states which do not have emission probabilities. So let us call them D. Okay, and the cost of a deletion will be the sum of going from the match states, M states to D states, and then there can be more than one D states, so you can go from one D state to the another, and that transition probabilities can be again trained so as to handle different kinds of gap penalties. So this process can give us all kinds of gap penalties. You can use an affine gap penalty, we'll train it in a certain way. You want to use even a complex function of gap penalty, we can do that. So any arbitrary gap penalty model you have can be trained by adjusting the transition probabilities of the delete states to the delete states within your Markov model. So here is an example of a basic match state with several delete states and the transition probabilities are given. Now as you go from one D to the another D, you can set the transition probabilities, thereby modeling different gap penalty models that whatever you may have. So this is the combination. So I have my basic match state model. On top of it, I have both the insertion states and the delete states. So you can move any which way. Now please note that once you move into a delete state, you again come back to the match state and so on and so forth. So in this way, any arbitrary sequence which may have gaps, insertions or deletions can be now modeled within our PSSMs. So this sort of hidden Markov model is called a profile HMM, and this is a very powerful tool. And once you have trained a profile HMM on a particular family of proteins, you can use that to search any other sequence, whether it belongs to that family or it doesn't. So again, for all such profile HMMs, the input is a multiple alignment, the same way as it is used in motifs or PSWM. Okay, so once built a profile HMM, we can use the Viterbi algorithm to determine the most probable state sequence within that complex alignment, which would actually give me the alignment of a particular sequence to the rest of the family. Alternatively, I can use the forward algorithm to calculate the probability that a sequence belongs to a particular family, summed over all the possible paths in the hidden Markov model. So in summary out here, we have discussed in pattern recognition several methods. Most of these methods, be it sequence motif, be it position specific weight matrix, be it even in certain sense psi-blast, what we have done, we have either used a multiple alignment as an input, or in case of psi-blast, we created that multiple alignment on the fly, and based on this multiple alignment, we created an approximation, a mathematical model, which might be a simple motif in the case of sequence motifs, which might be a probabilistic, a frequency-based model in PSWM or PSSMs, but without handling gaps, or which might be a probabilistic model which is not only the PSSM but also with gap penalty and insertion handling capacities built in. Then we have a profile HMM. So all of these, starting from using the multiple alignment, then finally creates a mathematical abstraction of the properties of the sequence family, which you can now use to search for more sequences as and when you get. So every new sequence you get, you pass it through these trained mathematical models, and depending upon their output, you can assign them to a particular family, which also means that you assign them to a particular function or a particular place where they occur, and so on and so forth. So a lot of biology now comes in. The moment you assign the family, a lot of biology is assigned to the sequence. So at the end of the day, these kind of techniques are very useful for directly relating a sequence which you might have obtained from an experiment to all the biology that is associated with the sequence, with the particular function or the protein or the gene it may be. Thank you very much.